Hi everyone, I'm Sehi from Harvard Medical School. In this presentation, I will introduce a grammar-based interactive visualization of genomics data. In the genomics field, there are many visualization tools reflecting diverse data types and analysis tasks. According to our previous survey, there are as much as 80 different genomics visualization tools. However, when we position them in terms of their customizability, you can see that many tools offer limited customization. In this figure, tools with more customization are positioned on the right, and tools with less customization are positioned on the left. Tools on the leftmost area are tailored for a specific problem. So if existing users get new tasks and data types, they need to find another tool for their analysis. On the left center, there are modern genome browsers, which are designed to visualize broader set of data types. However, they are template-based tools. This means they provide a limited set of track types. In such tools, many designs are already fixed by the developers, and only a few customization options are exposed to users. This makes it challenging to optimize visualizations for new tasks. For a motivating example, we previously developed a web-based genome browser called HiGlass. It was originally designed for genome interaction matrices with some additional basic visualizations like line chart, bar chart, and gene annotations. However, as the HiGlass community had grown, new visualization needs evolved. This made us to implement new HiGlass plugin tracks because existing tracks supported in HiGlass were not able to fulfill the new needs. And this implementation process were time-consuming and laborious, especially because we had to use low-level libraries like Pixie.js and JavaScript. We had the HiGlass infrastructure, which allowed us to reduce many implementation effort, but still, we had to write 1,000 lines of code to develop a single track. In both genomics and general visualization field, several expressive tools have been introduced to overcome such the limitation in the template-based tools. With this new y-axis, tools are positioned in a way that genomics tools are positioned on the top, while more generic tools are positioned on the bottom. You can see that on the rightmost area, there are some low-level tools like D3 and Pixie.js. While these tools are highly expressive, they are very limited in terms of ease of use. It takes many time to learn and use this tool to implement new visualizations. On the right center, there are high-level tools that balances between ease of use and expressiveness. While they are great tools for quickly creating custom visualizations, they still have several limitations when creating diverse genomics visualizations. For example, genomics tools like ggBio and Circus only offer static visualizations. Therefore, dynamic exploration of large-scale genomics data can be limited. More generic tools like Vega Lite and ggplot also have several limitations. First, they are based on tabular data format. This means genomic experts need to first manipulate dataset prior to using them. They also do not fully support a wide range of genomics visualization. For example, radial visualization are very common in genomics field for showing connectivity information between distant genomic positions. However, you cannot create such a visualization in those tools. Finally, some of them are limited in terms of data scalability, which means it is not feasible to efficiently visualize large genomics dataset using those tools. In our work, our goal was to design a visualization grammar that is accessible for genomics experts, but at the same time provide a highly expressive generic visualizations. To achieve this goal, we developed Gosling, a scalable and expressive visualization grammar for genomics data. This figure summarizes the design space of Gosling. Gosling enables to create diverse visualization types, including heat map, bar chart, line chart, scatter plot, and arc diagrams. And these visualizations can be easily converted into radial visualizations. This makes it easy to create a visualization like circus. Gosling also allows to create coordinated multiple views like matrix visualization with overviews. Beyond just the general visualization types, users can create more complex glyph-based visualizations like ideograms and custom gene annotation tracks. When designing the grammar of genomics visualizations, there were several challenges. First, the design space of genomics visualization is large. It not only includes any general-purpose visualizations like scatter plots, line chart, and heat maps, it also includes conventional visualizations like gene annotations and ideograms. Given the design space, we had to properly identify the diverse genomics visualization in the wild and reflect them in the grammar. Also, the genomics data is diverse and large scale. This means important pattern can arise from multiple genomics datasets at multiple level of scale and across many different locations. Therefore, the grammar should be able to handle large and complex datasets efficiently and allows users to create visualizations that are effective for multi-scale and multi-focus exploration. In Gosling, we adopted the following approaches to overcome the challenges. First, we used the concept of the grammar of graphics. This basically uses abstract visualization components as building blocks, such as marks, scales, and visual channels. We also designed Gosling based on our previous survey wizard and its taxonomy for genomics visualizations. This allowed us to identify the design space and also develop our grammar reflecting the actual visualization needs in the wild. To efficiently handle large and many number of genomics datasets, we use our high-class infrastructure. To allow multi-scale exploration, we supported semantic zooming. Using this approach, visualizations that are more proper for given zoom level can be defined by users. Lastly, we provided coordinated interactions such as zooming, panning, brushing, and linking. This allows effective navigation and concurrent analysis of large-scale genomics data. In the following slide, I will introduce the grammar of Gosling with actual examples. In Gosling, each track displays a single dataset. Using the concept from the grammar of graphics, users can use mark like rect, point, line, and visual channels like X, Y, and color to make custom visualizations. The example in this slide uses the BigWig file to show a heat map using rect mark for rectangles, including genomic position on x-axis and filling color with quantitative values. Because Gosling uses abstracted visualization component, it allows to easily change visualizations. For example, to draw line chart instead, users can use line mark and then map quantitative values on y-axis instead of colors. Similarly, users can change the mark type to point to draw scatter plots. Sometimes it is necessary to overlay multiple tracks. In Gosling, there is a property called alignment for this purpose. So using overlay option, users can superpose multiple tracks, here line chart and scatter plot. This allows to create more complex glyphs that are commonly used in genomics visualizations. This example overlays text, rect, and triangle marks to create ideograms that represent chromosomal structure. Using the similar approach, users can create custom gene annotation tracks. For example, this figure shows re-implementation of gene annotation tracks using Gosling based on existing tools like HiGlass and IGV. Gosling allows to use nested format of multiple tracks. Using the nesting, users can create an ideograms and juxtapose stacked bar chart. This allows users to see the chromosomal context while exploring some experimental data sets. As radial visualizations are very common in genomics, Gosling allows to draw circular visualizations. This is possible by only adding a single line of spec to specify layout. Whenever users change the layout option, visual marks and encodings are transformed to generate corresponding radial visualizations. Beyond just static encodings, Gosling allows to use coordinated interactions. In this example, users can use LinkingId properties to lock the scale of x-axis between an interactive brush and another view. This allows to set up an overview and local detail views. Last but important part of the grammar is semantic zooming. As mentioned before, it is important to make visualizations adaptable based on zoom level. For example, users can create regular sequence plot that shows individual nucleotide using text labels. However, because such information cannot be shown in detail in the lower zoom level, users can make such information hidden when zoomed out. This can be done by specifying multiple tracks overlaid on top of each other and defining visibility options. Here, users specify two tracks, one for stacked bar chart that shows the distribution and the other for text labels, and define a visibility of text so that it can disappear when there are no enough space. Here are some more examples using Gosling. This one uses 3 views for overview plus detail views. The view on the top shows overall information of structural variance, while the other shows local regions. Using 2 brushes, the user can compare details while maintaining global context. This is another example of semantic zooming. Here in the highest resolution, we are showing individual genomics variant using lollipop glyphs. As we zoom out, these glyphs turn into simple dots and eventually transformed into stacked bars to show hotspot and the distribution of different genomic variant across regions. Finally, Gosling scales to billions of data records by building on top of our existing high-class infrastructure. This one shows two genomic interaction matrices for the comparative analysis. All the examples that we showed in this presentation are available online, and you can also build your own visualization using our web-based editor. We also have documentation and tutorials where you can more easily learn to use Gosling. Also, if you want to learn more about the scientific background of Gosling, including design principles behind the grammar, please read our preprint. Thank you for listening, and I would be happy to take any questions.